Hello.
Hi.
Hi. Perfect.
Okay, great.
Yes.
Seems to work.
Yeah. Perfect. Chat. Let me put the chat.
I can see that very well.
You can see it?
Yes.
Let me try. Can I don't know, it's giving okay. It's quite slow in changing, but okay. I have to.
Yeah, that's fine.
Yeah, it's okay. Perfect. I put the chat on the side in case there are some questions.
So you don't need to do that, so because I can tell you.
Okay.
I didn't have that on the video recording. I would just
No, that's better now. Because then if not everyone that will go for tourism will say I went to see my family, and there will be a lot of cheating, which is not good.
Okay. So maybe let's start while I'm still letting people in. So welcome again to our lecture. So I think it's the second last lecture, but I have to look at the calendar while during the lecture, and I'll let you know at the end of the lecture how many lectures we still have. But at least next week we'll still have one. And today we have a special guest again, which is Fabrizio. And Fabrizio is a senior PhD student in the group. And I'll tell you now in a second why we have Fabrizio here. So this lecture we started talking about introducing methods from how to theoretically deal with stochastic processes, how to understand theoretically tackle non-equilibrium stochastic many particle systems, and we introduced methods from field theory, renormalization, and so on. And then in the second part of the lecture, I showed you some tools about how to detect low-dimensional structures, so order, in high-dimensional big data sets. Yeah, and now we're in this part of the lecture where we'll bring all of this together. And I brought in Fabrizio because his project is actually a very nice example of how the topics that we dealt with this lecture can work together. So Fabrizio is working on genomics experiments in the context of epigenetics. And he's dealing with, so to say, the processes that are going on in the embryo when the cells that are in the embryo make their first decisions. And he'll give you an introduction later. And but what Fabrizio actually does is he takes these large data sets and actually uses tools from field theory to understand these data sets that we now have in biology. And he developed for this, he developed field theory methods and together with differential geometry methods to understand and to deconvolve these experiments and to apply these experimental technologies now in new contexts. And I think a very nice example of the combination of the different topics that we had in the group. And so Fabrizio has prepared some slides and some notes. I have to warn you because Fabrizio's handwriting is terrible. He's a brilliant physicist, but with terrible handwriting. But don't feel ashamed just to shout if you can't read anything or if you don't understand anything. Just shout. And because what Fabrizio will be showing is a real science. So first, there's benefit to that, and this benefit is that you can actually read it. If you go to the website of the group and look at publications, the first publication on top will be exactly what Fabrizio is showing today. And the downside is that in reality, things are always not very pedagogical. You know, things are a little bit complicated. And so in the future, for example, the action will not look as nicely as the things that we discussed earlier in the lecture. And the theory will not be as simple as the simple epidemic model we were looking at. Yeah, that's the downside if you're looking into some real science. And but enough said about this. Over to Fabrizio, and I think I'll turn my video off. And then over to you, Fabrizio. And probably Fabrizio won't be able to show everything today. Yeah, so we will take part of the next lecture to continue talking about this.
Okay, perfect. I'm here. Okay, thanks a lot, Stefan, for this very nice introduction. Yes, I was again gonna warn about my handwriting, and it will not be. I have some slides, so I mean, at least I will try to cover it as much as possible. So, as mentioned, today the topic will be scaling in genomic data. So we're gonna basically try to mix the things that you learned in the first part of this lecture and on the second part. And in particular, I want like to start of my interest. So what put me into this project, and what I would. Okay, yes, perfect. And so this, I think, is this picture is for me one of the most fascinating, probably, questions that we have nowadays, both in biology, but as you will see, will turn out to be even a physics question. So, and the point is, how do the cells collectively build functioning organs and then organisms as well? Because if you think about it, at the beginning, we are just one single cell. Then these cells will divide to cells, 4 cells, 8 cells, and then they will become an organism. But if then you think about it, every cell during this time, of course, during development and later, must take a very precise, must be tightly regulated, so must take a very precise decision, not only in time, so during development, so when to take a certain decision, but even in space, so where to make this decision. And when we think about it, this is the organism is so messy, you know, it's because we have many cells that may interact with each other, then interact with the environment, and still we have a very nice organism may be formed by these collective processes going on. And of course, when we talk about cells decisions, we can think of it in basically 2 big categories. One is, for example, cell division. So when and where does a cell divide? And one is when and where does a cell differentiate? And when what we mean by differentiation is that a cell become another cell type. So for example, this is what I'm going to show in the next slide. This is the first, for example, the first called cell fate decision. So the first differentiation, if you want, of cells during development. Now, when you here, this is if you will ever face a genomic data, you will always find this is this E2.5, E3.5 that I'm highlighting now. This means 2.5, 3.5 days after fertilization. And as you can see from, we just for now disregard the first part, but if you start to look down from epiblast, these are called pluripotent stem cells, in the sense that this cell can become every cell in our organism. These are very, let's say, primordial cells, because as you can see, they are start to They start to form around 3.75, 4 days after fertilization. And these cells are extremely powerful because they really can become everything that you want. But from 3.5 days to roughly 7.5 days after fertilization, at 7.5 days, these cells make a decision. So they start, let's say, the first decision, and they decide whether to become mesoderm, endoderm, or ectoderm. And we are interested in particular, so I'm interested in very early development. So, and what are the, and what is, sorry, the molecular basis of this cell fate decision? Of course, there are many, many factors that involve in cellular differentiation, cellular decision making. The first factor that comes to mind is gene expression. If you probably have heard about it. So some genes, of course, we have thousands of genes in the cell. Some genes can be more expressed, some low expressed, and this will determine which kind of cell is the cell. But the cell we are looking at. But there are many other factors involved in this process, and in particular, one that I'm really interested into is called DNA methylation, which methyl is like, yeah, the methylation methyl that you know. And what is DNA methylation? So if you have no idea what DNA is, I will just say, okay, let's imagine the DNA just as a strand. Of course, it's a double strand that is wrapped around from Watson and Crick, but generally is formed by what is called base pairs, and they can have, yes, you can, I hope, yeah, I think you can see what I'm drawing. And along this DNA, you have different base pair. They can have different name. We call it cytosine, guanine, A, T, and you can have every possible sequence that you want. And on the other strand, you always have often, you have the other one. So you have C and G, GC, AT, etc., etc. Now, what happens is that when a cytosine, as you can see already, this wanted to be a G and it became a C. Oh, sorry. When a cytosine, this one, is close to a guanine, CG, the cytosines of this group, that we'll call it CPG, can be methylated. It means can obtain a chemical modification. Now we will not take into account what kind of particular, the chemistry of this modification, but let's say this can take a value that is, if you want, on a very basic level, 0 and 1. So chemically modified, so methylated or not methylated. And these, there are exceptions, but generally happens only when at the cytosines when it's close to a guanine. So as you can see here, AT, they will never happen methylation. Then you can have maybe later another cytosines close to a guanine, but maybe this site is not methylated, and so on and so forth. And why is this methylation important? Well, during early lineage decisions, or during the first decision in the cells, because there are huge wave and drastic changes in the levels, in the global levels of DNA methylation in the cells when these decisions are made. As you can see, what happens is that at 0 days, so at the fertilization, the DNA is written roughly here, we have 80–90% methylated. So why is this so? The reason for that is that we inherit the DNA from our mother and father that is already methylated. And what happens is that there is first a huge wave of of demethylation. As you can see here, we demethylate, we go from 90 to roughly even less than 20%, roughly 10%. And then from 3.5 days after fertilization to 7.5 days, roughly, we have this huge wave of de novo DNA methylation. And this is actually what I'm interested into. And so the main question will be, okay, how are these DNA methylation patterns established and then maintained? We know there are some enzymes called DNT3 enzymes that we are gonna talk later, that basically they go around here and there, they attach to the DNA, and then they methylate, or not methylate in case, the DNA. It's very complex, the process of how these enzymes actually methylate the DNA, and I'm gonna skip like all the chemical part about it, and I'm just gonna show you the mechanism of this process. And of course, sorry, one thing to mention, if you're maybe not familiar, sorry, is that when I talk about 80% or 20%, whatever percentage, I mean the percentage of methylation with respect to all the base pairs that can be methylated. So this is not 80% of the base pairs of the DNA, but only 80% of all the CpG base pairs. So the one that can be methylated. And this, I think, it's an important thing to stress out. Now, what we would like to see then is how we would like to see this wave, this huge wave of de novo DNA methylation, and so to get some information about this wave of de novo DNA methylation. And we will likely we have our experimental collaborators that did a very nice experiment. I will not enter too much in the detail of this experiment, but it's called a 2I release experiment, in which basically you culture cell in some condition called 2I condition, then you release into what is called serum condition, and these cells, they undergo exactly this transition. Exactly, the transition that these cells undergo in vitro has been shown to recapitulate the transition in vivo. So what happens is that what our experimental collaborators do is that during this release, so when they are released into serum condition, they, during this wave, they just take some cells out of this, they extract them, they extract the DNA out of this, and they can read the DNA, the methylation along the DNA. So basically we have very nice one-dimensional line in which we know, okay, this side is methylated or this side is not methylated. And then, because then you take out some cells at one time point, and then after some time, after 30 minutes, 1 hour, you take other cells out. And so you get another information. So here we have interesting information on two sides. One is the spatial information, because we know along the DNA whether the sites are methylated or not methylated. And then we have a temporal information. So we know how this methylation is changing over time. And this is a very nice, this is like a picture here on the right of how this experiment would look like, or what is the data that will look like. And I will do it. Okay, if I can change it, perfect. So what happens is that this is really, let's say, a very toy picture of how the data set will look like. So we have this, for example, we extracted 3 cells. Of course, there are in the experiment many more cells are extracted. The DNA is extracted from many more cells. So we have cell number 1, cell number 2, cell number 3. And we know what the CpG in these cells are. So the one that can be methylated. And for each cell, we have a reader. So we can say, okay, this cell number 1 at position X here is methylated, while cell number 2 and cell number 3 are not methylated. But sometimes what can happen is that some cell, or maybe some CpG of some cell, we have actually no information. This can come from the fact that basically these are very, very precise and technical experiments. So some fragments of the DNA can be lost, and so we can basically lose the information, and we don't know anything about. We know that this CpG should be there, but we don't know. We don't have any information. We don't know whether it's methylated or whether it's not methylated. So it's basically not detected. Now we would like to construct a data set given this information that we have, and even the controversial information, which are sites that we don't know anything about. And what we do is called an unbiased tiling. In the sense, we tile the data set and the DNA such that there are 10 informative CpGs per fragment. What I mean by that is that, as you can count here, we have, if we start to count, sorry, we have this first CpG that we know everything. For every cell, we know what is the methylation status. Of the second one, we just don't know in the third cell, but it's informative because we know that in the first cell is considered not methylated, in the second cell not methylated, in the third we don't know, but we consider it an informative CpG. And we take 10 out of them, and as you can see here in total, there will be 11 CpGs, but only 10 are informative because we have this one here, where we don't have any information at all in every cell. And out of these cells, what we can count is we can count number of what we call positive counts, which are basically the cell where the experiment tells us the result, okay, this is methylated. And if we count them, these are 13 positive counts. Then we can count the ones that are not methylated, and these are 9 negative counts. And then we have 8 that I would put this sign because we have no idea of what is their methylation status. So Now what we would like to do is, okay, let's compute what is the average methylation on this fragment. And we can, of course, these procedures must be repeated for all the fragments that we divide the DNA into. And a definition of the average methylation here, I will not enter into the detail of why it's chosen this, because you will see it's a little bit maybe counterintuitive, but we can call the average methylation of this fragment as P, which is the number of positive counts, plus 1 divided P plus N plus 2. You can already see the reason for that, why we have this plus 2 and plus 1 up, is that let's imagine that for some reason we took a fragment without any information. Then if we don't have this plus 1 and plus 2 here, we will have P0, Pn 0, so we will have 0 divided by 0. So it will be a problem. And so we take this definition of average methylation. There are other possible definitions, but for now we will take this one. And so now we read the data, we decide a way into which we divide the data set, we count the average methylation for this region, which we call it unbiased. And what we can do now is to read them. And this is what I'm gonna show. This is an overview of what we are gonna do. Is because we started here, in number 1. So we got information. Now don't look at compaction. Now we'll just look at methylation here. So we get our experiment, our sequencing experiment. We extract the methylation patterns. We find it in methylation patterns in space and in time. And then what we would like to do, we would like to process this data, do some compute some interesting observables of this data, some interesting statistical quantities. And from there, we would like to pass and try to infer the kinetics in sequence space. So how do these enzymes bind to the DNA, methylate the DNA? And what are the results of this binding? Because probably there can be something very interesting going on. Of course, here you already see what the interesting consequences that we will have a very description of this process that will be in what we'll call the physical space, but this I will define it later. So up to now, let's just focus on the first part, so the sequencing experiment and how can we infer the kinetics in sequence space. I think I will only be able to cover this first 2 steps in this lecture. And the first thing that we do once we created this data set is, okay, we save it somewhere and we can read it as with the command fread. In this lecture, you will only need these 2 libraries, which are called data table that you already know, and ggplot that you as well already know. So we read a file and we put it into the T, and we call it the T. And then this is how a typical data set of this kind of experiments would look like. So here on the left is called feature. Feature is, you will see later exactly what I mean by feature, but for now is unbiased. So it's the way we decided to tile the genome and to compute average methylation. Sample ID, as you can see, has this weird format 00HR00. This is the time, so when these cells are extracted. Start and end means the start and the end of the fragment, which means the position. So this, for example, this starts at position 1 and ends at position what is 3 million, no, yeah, something like 3 million, say 1,773. And so this is the width, is just start minus end. The strand we don't need in this point. And the CpG density is basically the number of CpG divided by the width. And this is, of course, this is NCpG is the number of CpG. And the coverage is basically the amount of information that we have. So the one that I was saying before, for example, if you remember, we have site that can be methylated, site that can be not methylated, and site that of which we don't know anything about. And this coverage that tells us what is the amount of information. So basically, P plus N, all both the positive counts and the negative counts, and will tell us basically the quality of the fragment that we have. And sorry, here I skipped these second names here is basically the chromosome. So where is chromosome 1 and this is chromosome N, then you have chromosome 2, etc., etc. Then you have this methylation called sec_monk, which is exactly what we computed. So p plus 1 divided p plus n plus 2, and then you have the error as well, which we're not going to deal today. Then you have, as you can see here, another way of which we can compute average methylation, because this is not the only formula. Of course, there are many different ways, but up to now, we will just stick with that, as I said. So now what we can do, as you can see, we have
maybe a little comment relating to last time's lecture, where I showed you this workflow for these experiments, in a different experiment. At this point, Fabrizio has already done all of these steps that I talked about last time. This normalization of the data, quality control of the data, and so on. So this is already at a highly processed data here.
Yeah. Yes. Yeah, that's true. Yeah. Sorry. This is the part that I will skip, as it will take quite some time. But yeah, this is already quite a very polished data set, as Stefan was saying. So as you can see, the first thing that we don't like, that I personally don't like, is to have the time in this format. Because, okay, it's maybe I want to do some mathematical operation with time and I don't want it to have it into this format. So what I would do is the first line here, which is called, we defined a new time, a new column that we call it time. And this, as you can see, it's a substring of sample ID. Now, sample ID has generally has this format, which will be, for example, 32 hours and I don't know, 30. This is the general format as you saw in the slide before. This substring tells us basically, okay, take whatever is in sample ID, the string that is in sample ID, and take of this the first and second element. So this part is taking this one and transform it into a numeric vector. Then take the second part, the fifth and the sixth digit of this string, which are these two. And this is, we're going to sum it, but how? Of course, these are hours and these are minutes. So if we want the time in minutes, we would like to do time is equal 32 multiplied by 60 plus, which is what I'm doing here, plus, in this case, 30. And these are minutes. Here, just divided by 60 for simplicity, but you don't have to do this last step. And then here they find a new column that is called wave, which will be the coverage divided by the number of CpG. So this is just basically the wave is nothing but P plus N. So the total number of information that we have divided by the total number of CpG that we have. And why is this also? Basically, this is the information per single site for each of these fragments. And as you can see, the data set now is the same as before. We just added two new columns. One is time and one is wave. So now time is a very nice format that we can work on. So now the second problem comes that, okay, we have this CpG density. This is, as you can see, we have, of course, this is a continuous variable, can take every possible value. And if we want to start to group together some fragments to make some global, to compute some global quantities, we would like to put them together, to group them together. So the first thing that we would like to think of is, okay, let's bin this CpG density. Again, CpG density is the number of CpG divided by the width of the fragment. So what we can create is we create a vector that we call it bins. As you can see, this is just as numeric. This is just the way you create a general vector in RStudio. This is sequence, means you create a sequence that goes from 0 to 1 by 0.05. So you will have 20 of these. So you will start from 0, 0.05, 0.1, 0.15, etc., up to 1. Now, it's very important. This is the way RStudio works. Maybe in other programs, you will not need this last passage. I personally don't know, but I know that in RStudio, you will definitely need this passage, which is you want to center the bin. Because in this case, values, for example, if you will have a value that is 0.04, you will go here to 0. Why Otherwise, in general, you will like, well, 0.04 is definitely closest to 0.05 than to 0. So in order to do this, we first center the bin. Okay, we just basically, we divide, this is defined as bins -1 minus different of bins divided by 2. Now, it's not important how the command works, but as you can see, we just created a new vector, which this will be just basically, if we call it A, B, this will be A plus B divided by 2, and etc., etc. This will be important later, we're gonna see. Now, perfect. Now we have our bins, we have our centered bins, and we would like to know, basically, to start to bin the data, to group the data together. And this is what we're gonna do. Oh, it's like, okay, perfect. This is done with this command, which is called find interval. So find interval will say, okay, take the column CPG density. So you remember that we have the column CPG density in the data table, the T, which was, I don't know, we have 0.023, etc., etc. And this says, okay, take the T, create a new column called CPG class, and this is the find interval of CPG density X all inside true. Now we are gonna see what is X is our vector that we create, our center bin vector that we create before, 0.035, etc., etc. This find interval basically is telling, okay, for each of these rows of the CPG density, tell me where does this value goes into, is in X. So for example, this 0.023, this function will give you the value 1, because this will tell you, CPG class will tell you, okay, this is 0.023 is in the first bin. And so you will get a new column, which will be 1, 3, 4, 5, I don't know, 1 again, etc., etc., which will tell you every value of the CPG density in which bin is. Be careful. So that's why we need to add a new command, because if you now look at the data table in this way, you will add some numbers here, but of course you will like this to be the value of the CPG density, the bin value of the CPG density. That's why we add this. That is very important. No, never forget to say, okay, now CPG class, I define it again as X to E of CPG class. This means take the vector X. Here we have, for example, 1. So X of 1 will give you the value 0.035. X of 3, so this will be X of 1. X of 3 will give you another value. And this will be our new CPG density prime that we will define it again as CPG class. I hope everything is clear up to now, and maybe even my handwriting was not as bad as always. And sorry, one thing is that sometimes if you want to, in this case, you can always do it inside the T, but here for simplicity, I just create a new data table that I call the T best. You're gonna see why. So this is our new data table that we created based on the T, where we add this new column. But in this case, it's better. Sometimes it's better to have different data table. Of course, you have to be careful about the amount of space that it takes. But if you work on at least this is how I prefer to work. If you work with small data table, just it's better maybe to have separated, and in order then you can see the differences as you work on. So if you do everything in once, then if you have to spot an error, it can become a little bit more complicated. If you work with very huge data table, you have to be very careful about creating new data table. In this case, yeah, likely they are not that huge. So what we see is that
I think you're usually using 700 gigabytes of
yes.
RAM on Qlik.
For this example, you can do it. What I generally do, no, you cannot do it. It's better just to have it to the same. Yes, I'm the one using terabytes. So yes, that's true. Yes, unfortunately, genomic data are generally, they occupy a lot of space. So you have to be very, say, clever and try in all the computation that you have in order not to waste days in computing, even a minute probably. So And here you see, here we created this very nice column that we will call it CPG class. Now, what I will do, this is here. Let's look at what do I call it stat. So what is stat is that I take this data table before that I called it the t-best, and here I just count the number of rows for each class defined as the unique combination of CPG dense, CPG class, time, and feature. So basically, this is telling me, okay, if you have a CPG class, for example, I don't know, 0.035 at time 0 for feature that is unbiased, so the way we compute it, how many fragments we have in total. And this will tell you, okay, this will be 742,130. And then you can do the same, so for unique combinations. So for example, for the CPG class 0.335 at time 0, you have 2017 fragments, and so on and so forth. So this will, if you look at this now, this data table, this new data table that I call it stat, you can see that this n is varying a lot, but too much. We go from 3 to 742,130. So we are really too many orders of magnitude of difference. So how is it possible? We have so few fragments that have the CPG class, and with respect to this, so what's happening here? And this is something that I always would like always to suggest, because here I made, of course, intentionally a mistake, something that is not, I would say, too precise, is that I defined the CPG class as beans that are evenly spaced. And what happens is that if you look at what is the distribution of CPG density, so in the sense you take all the fragments, and for each of them, as you saw before, we have the CPG density, and we make an histogram of this, you will see that actually there are no fragments that have basically a CPG density greater than 0.3, basically. So there are no fragments that have a CPG density greater than 30%. So what happens is that what we did is that we evenly bin here. Okay, this was not an even bin, but. And so we are so of course, the first bin will have all the information, the second one a little bit less, and then not anymore. And that's a problem. That's what I will, this is like my, would say, a suggestion that I give you. Every time you have a new data table that you don't know much about, just start to look at histograms. They will not take much time, most of the time, but you will just want to see how the data looks like. And as you can see here, we already spotted the fact that, okay, this CPG density, actually, they don't go from 0 to 1. They are not from 0 to 1 even in space, the fragments. There is not a uniform distribution of this. The distribution is definitely not uniform. It looks like very skewed distribution. So can we do it in a more clever way? And especially if you look even in log 10, of course, as you can see, basically most of the CPG, here I wrote the command that you need in our studio just to make an histogram. You don't even, for this very simple, you don't even need to use ggplot. You can just, for just a quick check. As you can see, this is roughly, this -2 is 10 to the -2, so roughly 1%. As you can see, most of the fragments have 1% to 3% of CPG density. So we don't want to define bins that are linearly and evenly spaced. And what we can do is that we use a function that is called quantile. And this is the command. Now we'll not enter into the detail of this function because it's quite complicated. But the idea is that you create, again, a vector as before. This is just as numeric. So it is a vector as a sequence, again, from 0.01 to 0.99. Again, the same division as before. But what we do is that we say, okay, we quantile the T of CPG density. So we take the column CPG density of the T, and this function basically will create bins in such a way that we have the same number of fragments, if you want, of CPG density for each fragment in each of these bins. In this way, you are sure that you create that the n that we saw before, so the number of fragments for each class, for this unique combination of CPG class time and feature will have roughly the same amount of information. And again, we center the bin as before. This is always to be done. And now, as you can see, I just apply everything that I did before. I just use these new bins, again, X, instead of the linear spacing before. And as you can see, now N is way better. We have a very nice, they are roughly the same order of magnitude of fragments per unique combination of CPG class, time, and feature. Now comes a little bit, I would say slightly hardest part. And what we're gonna do is that we have this data table DT-best that was basically all, for all the fragments, the chromosomes, average methylation, everything. And we have this stat that tells us, okay, for each of these combination, we know how many fragments we have. And here you learned it in the previous lecture, we can use the function merge to put these two data tables together. So we combine these two data tables by what we call key columns, so which are called CPG class, time, and feature. Sometimes you can use merge without specifying this by, because it understands it on RStudio understand on its own, which are the common columns in this two data table. But at least as I prefer, of course, if you have to buy 11 columns, then better not to put it. But sometimes I prefer just to specify them because I know what I'm doing at each time, at each step of my program. This is, at least for me personally, I think it's clear. Then of course it's your decision. If you're definitely probably better programmer than I am, then you can do it all in one line. And what you can do then after, you combine these two, you create a new data table that we call the tmean. We filter out region with not many data points. As you can see, this n down here, we say, okay, we take only n that are greater than 1,500, and be careful, here we are filtering out regions, so we are taking away regions that they are not informative, that we don't have much information on. We are not removing outliers. That's a very key point that I want to make. So never ever remove outliers in your data set. So if you have something that, if you don't know the reason why you're doing that. So if you have a rare event, for example, or a data that, okay, this, I know they are all centered around one AIF 5, you have to think about what this 5 can come out. It can come out of the biology, of the physics of the problem, or can be just a technical issue from the machine. Here we are just removing things that are, say, data that comes from a technical problems that we have, given that this machinery may lose some fragments, and so we have regions that we don't have much information. Then, now this last step, I'm gonna do it later. And yeah, so what I define is that I define after I merge them together, I filter out. Now I create a new data table that is called basically the tmean. I'm already creating it here. This is just the last part, the last part. And as you can see, when you already saw it in this lecture, when we have this format that I'm highlighting now, it means that this data table is going to shrink a lot, because here we are averaging parts together, fragments together. This is why we define the CPG class. And how we are averaging them, we are defining an average methylation by a unique combination, again, of CPG class, time, and feature. And we define as this average methylation. So we are average the methylation of all the fragments, but we do it in a weighted way. That's why I defined before the column weight, because this is telling me, okay, just don't average together fragments that carry less different amount of information. So give more importance to fragments that have more information, which we know more, and less to the one that we have less information. So this is why I use a weighted mean of, this is the p plus 1 divided p plus n plus 2. This is methylation segment. And waves is, again, how do we weigh the weight of the weighted mean, which is, again, divided by the amount of information per CPG. This is p plus n divided by the number of CPG. Then we can compute, again, the standard error, and we can compute n again, which is the total number of samples. This is is simple .n. And this last is called an AI.omit. You can often do it when you just remove rows that you have some values of an A where basically you don't have anything. And now this is how our new data, the tmin, that is the one where we computed just now, looks like. Oh, this is much, say, I would say, much more beautiful, much shorter, and of course, we just take few kilobytes in the memory. As you can see here, we have a unique combination of CPG class, time, feature. Again, this is unbiased, the way we compute the average methylation. We have this average methylation. So for all these fragments that have this CPG class at this time, we have standard error and the number of samples. So what we'd like to do, the first thing that we have, given that we have such very small data table, just plot it. We use ggplot. You know probably now all the nice feature that has that this plotting program has. And as you can see here, we plot exactly the previous data table. On the x-axis, we have time. It's this is, sorry, it should be minutes. There's a mistake. And no, sorry, in hours. It's correct. And on the y-axis, we have the average methylation, the one we computed before. The color stands for different factor in different CPG class, and the shape you're gonna see it later. And of course, then this is, I will not enter into detail of this code, just to make it look better, but it's not important. But what is important is that if you see all these curves that have different colors, these, they seem pretty similar, or at least to me, they seem pretty similar. The only different, the first thing that catches our eyes is that, okay, they have definitely a different initial value, a different offset. But maybe if we can get rid of the offset, then they will look even much more similar. And so when we think about similar, we are basically saying, do all these curves have the same functional form? So basically, we want to do what is called in physics a scaling ansatz. So we say, okay, probably these curves just are the same curve. So basically, the average M is just an initial value. Of course, we know that it's different for each of these CPG class. So this initial value, but they have the same functional form in time. So this is plus time to some power alpha. This is called a scaling ansatz, and here I forgot, but here a k, which is a prefactor. Of course, this can be different for all these curves. And how can we test whether this scaling ansatz is true? Because up to now, this is only an answer. We are saying, okay, maybe all these curves are the same curve that have this form. But is it true? And so then we have to test it. How can test it? This is just very quick code on basically how to do it. The first thing here is we create a new data table, which we call it dtmin collapse. Well, already this is a spoiler, but you're gonna see what's happening. And so we take dtmin, we create a new column that is called slope CPG class. And this is called LM. This means a linear model. Then this is weird because we are applying a linear model to something that is, as you can see, not necessarily linear, but this actually works because linear model basically works for y equals some function of x, and it just gives you the intercept and the slope. So, and so basically we say, okay, here is an important point to stress. There are different ways, and probably if you look at, if you've done some courses on statistical physics, you saw the Ising model, and when you want to find the critical exponent numerically, you compute different system sizing, et cetera, and you try to collapse all this curve into a single master curve. And what you do is just basically you try different value of the exponent till you find the one that best collapses the data. Here I already, I tried, of course, different values, and I found that the best value will be 2.5/2. Now you're gonna see what I mean by best value for what, but you're gonna see that this curve will nicely collapse. And so what I define is that, okay, this first slope CPG class will basically find, based on my ansatz, so my ansatz is average m is equal to m0 plus k to the time to the 5/2. So what this function again can find is only m0 and k, but you have to give the 5/2. So you have just, again, as I said, try different values. And this first line will give me the value of k. So will give me the value of k, as you can see, by what? By CPG class and feature. So for each CPG class, so which being CPG density, for each features, in this case, we have only the unbiased, we are gonna find a value of k. Perfect. Then we have the second line. Okay, now we want to know what is, as you might guess, we want to know what is the value of the intercept. So once we know, we don't even know to know k, because this function will just give you both m0 and k. As you can see here, k you extract as lm, you have this 1, 2. This is basically telling, okay, lm always gives you both m0 and k. This is just say, okay, extract k, and this just saying extract m0, so the initial offset. So now what we can do is that, okay, first, let's just take the average methylation and remove the m0. So this is done. We define an average methylation prime, which will be just average methylation minus m0. And we hope that this will be kt to the power of 5/2. And now we say, okay, in order for this curve to look all exactly the same, now this k, we have to include this k somewhere. And what we do is called, basically, we rescale time given k. So what we are going to do is that now we have this average m prime is equal to k to the t to the power of 5/2, and we defined a t prime as k to t k. This is my mistake. It should be okay. 2/5 dot t. This is what I have done here. This is k to the power of 2/5, so 1 divided 5/2, okay, t. So this is my new time. And as you can see, with this newly defined time, now we have that for all of this curve, we should have something like this. Average and t prime will be t prime to the power of 5/2. And well, if we now just plot this, the t-mean collapse, we can see that all this curve nicely will collapse into a single master curve. So we were able with this 5/2 to collapse all this curve into a single curve. And again, we know that we found out that the average methylation increase as the time to the power of 5/2, which is, of course, already something quite striking is coming out, because this 5/2 is not a very common, I would say, exponent for an increase over time of any quantity. Now we'll just, what I have, okay, which just did 50 minutes. I will just now maybe go briefly into that and just say what we have done so far. So our workflow was, we computed the average methylation in regions defined by this unique combination of CPG class, CPG density, now we call it CPG class, which was the bin, and time. Then we found this very beautiful, that we use a scaling ansatz, and we found at the end, by using the scaling ansatz, that all these curves basically are the same curve, just with a different case on this slope, if you want, sort of slope and initial offset. And so we found that the average methylation at the end increased as alpha plus some power of time, some function of time, and this function of time is t to the alpha, with alpha equal to 5/2. We check the best value. Of course, what you can do is to try to look, because of course here I put a power law as an ansatz. You can look at different ansatz. Maybe it's an exponential, maybe it's linear, maybe it's a polynomial. Of course, just be careful with polynomials. And you can check all of this and find, well, what is the best one to collapse this curve? And we saw that basically the best one. Of course, if you go to a polynomial of 200 order, it will be the best, but that's always the case. So for reasonable function, actually the power law is the best. It's the best function to collapse all this curve. Now we find this, we define everything based on this unbiased. So we divided, if you remember, our data set, we tile it into regions with respect to the 10 informative CpGs. But sometimes maybe we want to look at how the average methylation is changing in time for different regions of biological relevance. For example, we have on the DNA what's called gene bodies, introns, exons, or promoters of the gene. And they generally, maybe like promoters, can be very short. So if we take, for example, 10 informative CpG, which will be this green line here, well, what you're going to see is that, and this red line, for example, is the red scribble is are the introns. Well, we are going to take many more CpG that are actually out of this region. So what you can do is that actually you have to not take only 10 informative CpG, but you have to take what is left. So in this case, you will have a region defined by just these 5 CpG, and you will have fragments that will vary a lot, the number of CpG probably, but you cannot do much about it because these are the regions that you want to look at. And you can do exactly the same procedure. Of course, you have to create a new data table, put this data table together, where instead we have a new, as you can see here on the column feature, here there is an unbiased was the one before, and now this is H3K4ME1. This is a new region of biological relevance. And so this data table, that's why they become terabyte of data, because you can start to look at many different regions like introns, promoters, H3K4, H3K27, so they will become huge and huger. But we can exactly use the same code as before, because we just add feature every time we had this unique combination. And as you saw before, I already had it. So if you want to use this code, basically, it's just, you can apply one-to-one. So I already had it before. So the unique combination will be not only of CpG class and time, but we add a new key column that we call it feature, which are all these different regions. And once we do this, we can do the collapse again, everything exactly like before. We just add this column feature. And as you can see, this is what happens. That's very, I will say, very, very beautiful. And all this curve. So as you can see here, we have so many, 20 CpG class. Then we have, I don't know how many here, there are 9 different biological regions. In this case, the colors are the biological regions. The shape of these dots are the CpG class. And as you can see, again, we are able to collapse all this curve into a single master curve. So there is the same mechanism of de novo DNA methylation in different regions of the genome. So now comes the part in which, okay, now we learned quite a lot. So we know, at least we know that how the average methylation is increasing with respect to time. So we can say, okay, let's try maybe to do a model, to build a model based on this observation. So first of all, every time we build a model, we have to think, okay, what do we know? What have we learned? So we learned that the average methylation increases the time to the power of 5/2 in every region. This already is telling us, okay, you don't need to create a specific model for each, for introns, for exons, for a region that you want, but you can create a very general model that, of course, then the scale, so this sort of slope will be different for each of these regions. But if we just want to find this time to the power of 5/2, this will be sufficient to have a very general model, so not, or at least a not specific model. So we know that DNA methylation is established by the NT3 enzymes, and 5/2 is not a trivial exponent. I would say it's not linear in time, and this is suggesting that there are probably some collective processes going on between these enzymes in order to bind and to methylate the DNA. It will become much clearer later, but up to now, we can think of it. Okay, so what is the simplest collective process we can think of? Well, we can imagine that there are some NT3 bounded on the DNA, and they just basically call to get other enzymes to bind to the DNA and then to methylate. And this process will be for each of the sites on the DNA. this process will be just D that goes in D plus D. So it means that if you had, for example, in this case, you had 3 enzymes, then they will call probably another enzymes to bind to them to the DNA. And this, of course, the rate of this process, we call it KD. Now, how can we translate it into an equation? And if you remember, we can use the master equation. So we know that how does the probability of observing a certain enzyme pattern at time t changes with respect to time. When I use this bracket, I mean, as you have highlighted here, this bracket stands for d1 to dn, because of course here we have what are called lattice position. So we have I equal 1 up to I equal n, where the lattice positions, again, they are not evenly spaced, but they are the CPG. So I'm not taking into account all of the other sides, and only taking into account the CPG, and I'm naming basically them from 1 to n. And this, again, this D, this bracket D means D1 to the n. So it means that, for example, if you will have a pattern like 10, 1, 2, etc., etc., it means that at the first CPG, there are 10 enzymes, at the second one, just 1, at the third CPG, 2, etc., etc. So how can we write this very simple first collective process of binding? Well, this is, so the T of P of D times T is equal to, if we look just for one single site, it will be like, and when you write the master equation, just think about how do I get there? So how do you get there? You get there if you add at one position D minus 1, and then you bind 1, and so you get to D. So you get D minus 1 per P, and this is, okay, let's add the position here. So at position I, this di means all the, again, all the vector excluded I, and so di minus 1, and this happens with a rate kd. So again, this is why, well, you can say, okay, this process is proportional. Again, if every D can call, every enzyme can call another enzymes, this rate will be proportional to the number of enzymes bound. So this is why it will be proportional to di minus 1. And y minus 1, again, because we want to arrive to D. So if we have, at the same time, we want to arrive to a vector 0, 2, 0, 0, 0, we arrive it from 0, 1, 0, 0. That's, and the process of binding will bring us here. That's why you have di minus 1. So once this is clarified, of course, this is for the lattice size I, so the CPG I, but this can happen on every site. So we have to sum over all the possible sites where an enzyme can bind. And then you have what is called the, sometimes I forget to put time. Actually, I will exclude it. If it's not necessarily for understanding, I will just, here is always intended time, but I not explicitly write it every time, because you will see that it gets much more confusion, and we'll get much longer equation. And now here we are always the loss term. So here is how do we get into probability to have observing a certain pattern, and then how do we get out? Well, in the same way, just that sometime, just that this time we have di p of d of d. And p of d, because this, of course, will be proportional to the number of enzymes bound, because each of these can call another enzymes, and so you will get to d plus 1. That's why you get out of this state. The same thing you can look for binding. Well, this is if we have a very similar process, but the binding is the opposite. So how do you get, for example, from to 0, 4, 0, 0, 0? Well, you get from here from 0, 5, 0, 0. Because this is, so when an enzyme is unbound with a rate U, then you from 5 to a position I, you get to 4. So if you want to write just this process as a in a master equation formalism, you will have U sum over I of, sorry, what is di plus 1. Here we'll have again P of d I di plus 1, and then you have minus u sum over I di. These are actually the same. These two are, they look exactly the same. They have a different rate. Now, then you have, we can add a spatial process, because up to now, everything here is happening, of course, can happen on different sites, but there is no communication between different sites. And here we can add, for example, what is called diffusion, if you want. So an enzyme that can slide on the DNA. And this will happen with a rate that we call it J. So again, how do we write this? We have always to think about how do we get to the state P of D T. How do we get here? Well, let's imagine, of course, these enzymes can jump, these enzymes can jump to the left or to the right. So we can get, for example, for a jump to the right, we go from di to di plus 1. So where this di is, of course, you have di enzymes here and di plus 1 here. And by just one jumping out and going from I to I plus 1, so at the end we will get one less in I and one more in I plus, at position I plus 1. So if we look just as this left jump, then this will be just, this will happen with a rate, J. And here we will get, again, di plus 1. Then we will get P of di plus 1, di plus 1 minus 1, and that's actually d excluded I plus 1. And then you have the loss term again, and then here again you have the sum over all I. This is for the jump to the right, jump to right, and then you have a very similar one for the jump to the left. All together, we will have a very nice, we can put all these different processes together and write down the full master equation, which will comprehend different terms. The first term is the P binding, then we have the unbinding, and then we call it diffusion if you want. And this is the jump to the right that we just found, and plus the jump to the left, and here is minus the loss terms. Perfect. Now I want to just, for one moment, to let you reflect about the master equation, because every time you you face a master equation, often in course, they will say, oh, it's generally impossible to solve the master equation to get an exact solution, and actually it's true. But if you think about it, the master equation is quite simple, because at the end it's linear. As you can see, it's a linear order differential equation. It's in P. Here you have P, 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 P everywhere. You don't have P squared or sine of P. That would be quite hard and nastier differential equation. So if we are able to write this master equation into this form, so the T of x equal alpha x, well, then we know the solution, we know the exponential solution. So that's, I would say it's always important to think of this in order to understand how maybe people that maybe wrote down field theoretical methods arrived to this, because probably they saw the master equations. Okay, probably if we are able to write it in this very simple way, then we can try to look for a solution of that, maybe a simpler solution. And how can we do that? That's the hardest part, because up to now looks not really in this form. Well, and here comes the little bit the nastier part of today. And in order to do this, I will introduce this called the Doi-Peliti Phi integral formulation of the master equation. Sorry. So, and first of all, I will define what is called a Fock space. And what we have is that, so what is our observable? Our observable up to now are a pattern of enzymes. Up to now, we are not even considering methylation. So this enzyme, we know that they can bind and they can methylate the DNA, but just let's consider simply the binding of the enzyme. So we have a certain pattern of methylation, and what I would define, this is just a definition for now. It's what is called the Fock space, and this is this weird symbol that if you have never seen, we call ket, which is ket of D, where this D stands for D1 up to dn, so will look like very similar to this pattern. And this is defined as the product over all the sides of some a dagger j to power of the j0. Now we're going to see what this operator do. These are called creation operator. And this 0 is the vacuum state. So for a Fock space, we just need a vacuum state, which means all zeros. So basically a pattern without any enzymes. And some operator that basically puts some mathematical object that basically puts an enzyme. And this creation operator, how does it work? Let's imagine that we have a pattern, so this vacuum state, 0, 0, 0, and we have a dagger 2. So a dagger at position 2, it means that we add 1 at position 2 of this ket. So here, this is the position 2, and we add 1. We add 1 here, so this will turn out to be 0, 1, again 0, 0. We'll leave unaffected all the others. We just say +1 to position 2. Then we can actually apply it 4 times. For example, here we have a dagger 1 to the 4 to this other ket. This is maybe it's a different, now we don't have any more the vacuum state. Let's imagine we have a general state like 2, 3, if you want 0, 7, whatever it is. This is what happens. Of course, if a dagger applied once will increase by 1, if you apply 4 times, it will increase by 4. So we will get 6, 3, and again, the other one will be untouched. But of course, you can apply this procedure to 2 different sides, because, for example, we can first raise by 1 the side in the ket, so the index number 3, and then the index number 1. So in this case, what we will get is that, let's imagine now we have this 2, we first 2 to the number 3, so from 7 goes to 8, and the first one from 3 goes to 4. And this is very, very simple definition. Now, as you can see, what happens if you apply this, if you write down this one explicitly, that's why it's the definition, because now let's imagine we apply a dagger 1 d1 times a dagger 2 d2 times over 0. So as you can see here, we increase by d1 the value, the index 1 that was 0 because it was the vacuum state, so will become d1. Then you apply d2, and this will become d2 up to the end. And that's why this will be defined as the simpler. Then in this Fock space, so this is happening now, I don't have to worry, this is just definition. Since that there is no clear relationship with the master equation, we are going to see the relationship later. Now it's just pure definition. If you maybe had a course in quantum mechanics, you heard about second quantization, which will very look like this. And here you have what's called the annihilation operator. So the annihilation operator does the opposite as the creation operator, almost the opposite. Basically, instead of increasing by 1, so raising the index I by 1, it lowers it by 1. But not only does this, but it takes out a number. So as you can see, if I apply A, so the low annihilation operator or lowering operator at position I, you get di, so you get a number out, prefactor out, and you lower by 1 the number that was at position I. So if you, for this example, if you have A4, so we annihilate the index 4 by 1, what we'll get is that the index 4 was 6. It is 6 here. So we get a 6 out. The other one are left unaffected. And this will become 5. 3, 0. And here I just, these are all the, again, this is defined in the same way as before. These are all, if you want, all the properties. So AI applied to D just gives out, gives a factor di by the vector. A dagger I never brings out a prefactor, but just raised by 1. And then you can apply it to the to what is called a bra. And so you can apply, if you want, this operator to this other, in the other direction, if you have what is called the bra here. and they will do basically the opposite. So AI applied to the bra will raise by 1 instead of lower by 1. I will bring a factor di plus 1, and a dagger I actually will do will just lower it by 1, as you can see here. This is not, we'll see, not too important for now. And this is, there are some very nice properties. One is called, that's why they are defined in this way, because they have to respect what is called commutation rules. So that AI a dagger j must be equal, the commutator of AI a dagger j must be equal to the delta of ij. And by commutator, I mean just a commutator of x, y is equal to xy minus yx. This, of course, okay, should be 0, but this, be careful, are not numbers, are operators, so it's not 0. And this is why now you will see they are called bracket, because when you have a bra here, d prime, which is another, another sequence of this, and you have a d, this is called, when you compute this, this will give a product of delta of d1, d1 prime, d2, d2 prime, etc., etc. So it means that this one is just non-zero when they are all the same. So when d1 is equal to d1 prime, etc. You just need one that is different, that this would be 0. Now we define another very important operator, or a combination of operators, actually, which is called the particle number operator, which is a dagger I AI. Because what happens if you apply AI, this will give you di. Let me do it maybe step by step. So when you apply AI, this will brings out, if you remember, a factor di, and will lower the d at position I by 1. Now, when you apply a dagger I, this will raise this one by 1. And so this will give you di d. So as you can see, this vector, this ket, sorry, not this vector, this ket has been left unchanged, and you just brought out di, this y. It's basically counting how many you have at position I. Now, let me just define another quantity based on this creation and annihilation operator. This quantity, we call it up to now phi t. So this is phi t, is just defined as the sum over all the possible value of d. So all the possible value of d1, d2, up to dn. Multiplied by the product of j of a dagger j dj to the power of dj, p of d of t, which is our probability distribution of observing a certain pattern at position t, and this vacuum state, which is this. You can rewrite it in a much compact form, as you just have to apply, as we showed before, this to this, and you can just write it in this way. So sum over all d of p of dt d. And now let's see why this will become useful. So first of all, let's apply this thing, the pink operation that you saw. Let's just apply the pink to the left of the master equation. So if you remember, the left-hand side of the master equation is the t p of d. So let's apply all of this. So you will have some sum over d product. Then of course you have to apply to the right-hand side as well. We have this a dagger j power of dj, the t p of d t. Then you have 0, and here again you have it. You can write it in this way as the t sum over d p d t up to d. Well, and as you can see, this is just the definition. This one, just phi of t. So we can nicely rewrite it at the t of phi. Okay, this seems hasn't done much. We just change from the t of p of d to the t of phi. So here nothing much has changed, but we have to apply everything to the right hand side as well. And let's look, for example, to the first term. To the unbinding at the beginning, which is easier. And when you apply to the unbinding, Of course, we apply this again, the pink that we had before, and you will have a term that would look like this. So, and in particular, let's just focus on this part. So what you have here is di plus 1, and then you have, let's write it explicitly, you have d1, di. Again, be careful, now we applied this one, we applied the pink one, so we don't have, even though this is di plus 1, we don't have di plus 1 here, because this is, and so this would be dn. So the point is, is there maybe a way to write this part in terms of creation and annihilation operator? Well, let's think about it. If we write a dagger I of d1 di plus 1 dn, well, this doesn't work, right? Because here we increase by 1, doesn't work. But what happens if we do the lowering operator? So if we apply ai to d1 di plus 1, this we know that will bring out a factor di plus 1, and then we'll lower by 1 the one at position I, so we lower by 1 di plus 1, putting it to d. So this d1 di dn. So as you can see, this one has been rewritten here. Yes, if we has been written in terms of raising and lowering operator, raising and lowering operator, as these two are exactly the same. So we can write this, all of this, simply as u ai times the vector d. And that's, of course, so of course, if you want, I mean, you just have to rewrite it in that way. And as you can see, and as you will see, once we have done this, well, now we can, we will have, again, the sum over I, sum over D of U, then AI, this P of D, D of, be careful, this will be D of I plus 1. Oops, this is my handwriting. And the point is that now this will be, again, you will have here a di plus 1. And this now, given that you have di plus 1 here, di plus 1 here, we can put it as a dummy variable. And so this will be simply the sum over I of U ai phi. What we can do is that now we can try to do the same procedure to all the terms of the master equation. So for example, now we applied it to the unbinding. Now let's apply it to the binding. Now the binding, it's a little bit harder, but again, let's just look at this term di minus 1 d. So this will be di minus 1, 1, 2, and we want to write it in terms of raising and lowering operator. So what we can try to do is that we can, let's just apply, for example, the first, this is the reason, how you can reason. How can I get di minus 1 as a number? You can get it if you apply ai to this vector, right? To di minus 1 dn. If you apply ai to this cat, sorry, then you will get exactly di minus 1 and this. But what will happen here is that you will get the right prefactor, but unfortunately, you will get the 1 di minus 2 because it will low by 1. So, well, if you think about what can we do? We get 1 less. We have 2 less of what we want. Well, just maybe apply twice the raising operator. So if we just do the dagger I square ai d1 di minus 1 dn, this will be exactly di minus 1 d, which is what we have here. in blue. That's perfect. So now we can, again, look at the I-1 will become an Hamming variable, etc., and we can just replace all this nasty expression here in terms of raising and lowering operator in a very nice and simple way, because this will just be some, if we just maybe this will become sum over I of a dagger I square AI over 5. Then you can, I mean, I will skip this. If you want, you can do it as an exercise. And you have the loss terms again. So the loss terms in the master equation. So for example, you had, if you remember, you had the terms KD minus in the master equation. There was like KD di P of D. You have again to apply this, all of this pink. So the product and the sum over all of D, and rewrite it in terms again of raising and lowering operator. Here you will see it will be much easier than the one before, because here you will have something unchanged, because this will be P of D exactly. And if you remember how to use the particle number, it's basically very simple. So again, what we did is basically, as you saw before, we just wrote all the master equation in this very, very, very simple way in terms of raising and lowering operator, but we don't have anymore a P of di minus 1 of P of di plus 1. We have just phi and some raising and lowering operator. But as you can see, if we just call all of this, put all of this operator and we call it H, and this would be a dagger I, this is what we computed, a dagger I AI squared, this is again what we computed. This is basically the loss terms, because as a convention, remember you put always a minus here. So basically here we computed as a plus, then you put a minus. So again, comes out as a plus. And so we rewrote all the master equation, which was again in terms of PDI plus 1, PDI minus 1, in terms of phi, and some raising and lowering operator that we put inside H. So we can write this master equation in terms as a simple linear differential equation. The T of phi is equal minus H phi. Well, that's very nice because we know the solution of this. Because we know that the solution is an exponential. So we know that phi t is equal to e to the minus h t phi times 0. Now, you can say, okay, can you just tell me the solution, given that if I know phi of 0, can you tell me what is phi of t? I will say no, because I don't know how this exponential of minus ht actually acts on phi 0. And then actually, I think I'm afraid we are going to see it next week. We are going to see how we can apply this e to the minus ht to phi 0 to get at the end phi of t. And again, if you want as an exercise, let's try to write the diffusion part, so left and right jump in terms of raising and lowering operator. You can find it in every standard textbook like CARD if you want the solution, or maybe I will give it next week. But I hope, so I think that for now I will have to maybe, no Stefan, because so maybe I can, we started a little bit later, maybe I can.
Yeah, sure.
5 minutes, if it's not.
Sure. Yes, I'll just to make a clean break, and then and then we can continue next time.
Yeah, perfect. So I will just give you this thing because up to now maybe everything is left a little bit in the air. So because we have this phi, but again, this phi was defined in this very weird way in terms of raising and lowering operator, some vacuum state, and so why is it useful? Okay, maybe we can compute this phi, as a e to the minus ht phi 0, but then what we get out of that, because it seems a little bit out with respect to the master equation. And then actually, for example, can we get the average number of enzymes bound at position I at time t? That this is the one down here. This is just the definition of the average number of enzymes at time d. So can we get this into this formalism? And in order to do that, I will do what is called, we introduce what is called the projection operator. The projection operator, which we call it pi, will just be the sum over of d of this bra of d. As you can see this, you can rewrite it in terms of the sum over d of 1 over d factorial to e to the d times the vacuum state. Now you have to remember why you have this d factorial, because if you remember when you apply a to the left of d, then here you will get d plus 1, d plus 1. That's why in order to have d, you have to divide by all the factorial d. And again, now nicely, 1 over sum over d of 1 over d factorial a to the d, you can then write it as an exponential operator. Now, if we, for example, let's do this. Let's apply this projection operator to the particle number of this projection. I call it projection operator, but maybe it should be, I should call it just projection bra. I have Gunnar Prusler calls it the abyss, because basically, but maybe I will just leave it a projection operator because it's maybe clearer. Maybe I will find a different name for the next lecture. And so what happens is that if you compute this pi, the bra of pi, a dagger I, ai, phi, this is just use the definition. This will be the sum over all of d. This comes from the sum over all these comes from this pi. The sum of d prime, if you remember, comes from, if you remember that phi was defined as sum over d, p of d, d. So this is, okay, so we will have the sum over, sum over all d that come from the pi, sum over all d prime that comes from the phi, and then you have a dagger I, ai. Now we have a sandwich between those. So let's first compute here. We can rewrite it in this very simple way. The sum over all d prime. We can take p d prime out, and we have to compute d a dagger I ai d prime. This will just give you sum over again d prime over p d prime. Again, if you remember, this was first you lower by 1, then you raise by 1, and just brings a factor di. So this will be di over d prime. And if you remember, this was a delta of d prime. And actually, sorry, of all of d prime. Delta d prime. And so this will kill one of the sum. And so this will be d d of d. And this is nothing but the average that we have before. So this is just the average number of bounded enzymes at position I. So now we have a way to compute an observable such as the average, and then later maybe correlation functions. Once we know phi, we just need to apply this pi, and that's done. And then we get the average value of enzymes, the correlation function, and all the observable we want. And yeah, I will think I will stop here for today. Next lecture, I think we are gonna see how then you basically solve the previous the master equation newly defined in terms of this raising and lowering operator in terms of phi, and then how to construct the field theory based on that. And then we're gonna wrap it all up and to maybe see how do we get this 5/2 out of all this mess. Thanks a lot.
Great, thanks Fabrizio. So I suggest we stay online a little bit just in case there are any questions.
Yep.
And otherwise, see you all next week. So the first part of the lecture next week will Fabrizio will continue. and reveal with a big revelation what is behind these epigenetic processes.
Yeah.
And so if you weren't able to read the handwriting, as I said, you can read the paper that's on the homepage. Okay, see you all next week. Bye.
Bye.
I have a question.
Yeah, sure.
So why, when you were doing the modeling and you said you put in like a low diffusion term,
Yeah.
but then you said that these lattice sites are not evenly spaced. So like
Yeah.
1 and 2 are not going to be, it's not going to be like a diffusion, right?
Yeah, exactly. This you can, if you want, you can extend it to a diffusion, like a jump to every possible site. That's you can.
Okay. So it's not just like neighboring site. It can go to some.
You can think about going to the neighboring one. At the end, we'll give you a spoiler. This model will not be correct. But
okay.
No, but this is just a spoiler. But the point is, you can go roughly through that. You can, if you want here, you can make it as complicated as you want, this diffusion term. And for example, putting here a j of I plus 1, and this maybe will depend on the distance between these two. So here you can introduce what is the actual distribution of CPG sites and say, okay, maybe if they are too far apart, it will be very unlikely that you will jump from one to the other one, for example.
I see. And like similar to that, I guess, you could have said, instead of like saying that you get a demethylation with some rate or something like that, could you have said if like 2 are there, then they like, I guess, like coagulate and
Yes,
get rid of one? Like how do you like decide, it like to get rid of methylation? How do you decide? There's so many choices that you can do.
Yeah, exactly. There are so many choices. Yes.
Yeah.
I definitely agree. And they are, this is, for example, okay, this was just for me a pedagogical way to get to the field theory. Then you're gonna see the correct model. But the idea is that, okay, let's just take the first simplest possible process you can think of. Because, for example, you can add, I don't know, in terms of coagulation, 4, like, for example, 2 enzymes together call 10 enzymes. I don't know. You can add all these possible processes into account. And this, of course, will make, and but then you have, once you add all of those, then you can add them if you want. Then you always have to come back to your data and ask, okay, does this model will predict our data? And then you are gonna see that actually whatever coagulation terms you will put, like this sort of coagulation on the coagulation, will not explain the 5/2, and you will need to use some fractional coagulation. So you will have to be very cheesy in doing it. So we will see that this will very simple binding, collective binding is not sufficient then but to explain the 5/2. But yes, this is for me was just really a very simple example to introduce the field theory to have very simple processes that we can write it in terms of raising a lowering operator. You can add all the possible processes that you want. It just be harder than to write it. Yes.
I see.
Sure.
So in physics, we always have, in principle, you can always have an unlimited number of models that describe the data with arbitrary complexity. For example, you can describe the motion of planets not only by Newton's law, but also by Newton's law plus a term that takes into account the color of the planets, plus a term that takes into account the temperature of the planets and so on. And these would be all right. They would be able to predict the motion of the planets. But in physics, we're always doing good when we consider the simplest possible explanation for something. Yeah, and that's also what Fabrizio will be doing here. Yeah, and in terms of this disorder, this I don't know if I understood correctly. So these positions of the CPGs on the DNA, in principle, you would expect that this is important. For general models, if you have like disorder, your rates depend on where you are, you would in principle think that this plays a role, but you would never be able to solve something like this analytically in general cases. What we did in this case, we checked using numerical simulations that processes like this or things like this disorder are not that important. Yeah? That the simulations, that we can actually predict these simulations that we ran using a simple lattice like the one that Fabrizio was talking about.
Yeah, exactly. And then if you think about it, for example, when you take all these different genomic regions, as I said, they have all different distributions of CpG and different, maybe some are very dense, some are very, very less rare, but you will still see this power to everywhere, and then we are going to see it even for correlation function. That's already tells you that maybe this distribution is not actually that important.
I see. Thank you.
No.
Okay, so if there are no more questions, then see you all next week. And thanks for Fabrizio for spending his vacation in Rome, showing us some of his stuff. Okay, bye.
Bye. Bye.